Good evening, everyone. So we'll be waiting for 5 more minutes until everyone joins and then we'll initiate the session. Good evening, everyone. Okay, I hope everyone has joined. Let's initiate it. So, okay, so I myself, Tanishka, so I'll be assisting you for this in the live session of this particular course, which is Human Physiology and taken by the Professor Nishikant Subedar from IISER Pune. Okay, so uh, okay, so in these human physio course, in last class we studied about the blood circulation from heart to the lungs and its circuits, its pulmonary circuit and the systemic circuit, okay, and how the heart does the uh, contraction and goes in diastole and systole, okay. We studied the rhythms and yeah, okay. Hmm. Okay, so in last class, we studied how the action potential is being generated in the heart muscles. Okay, so as we have studied in the first class, also we have skeletal muscle, we have smooth muscles, we have the cardiac muscles. Okay, so these cardiac muscles have a different kind of mechanism. They can generate the action potential from themselves. Okay, they are autonomous for generating the action potential, and due to that action potential, the heart goes the continuous rhythm of systole and diastole, and that is how we get our heartbeats. Okay, so we read, so here we can see that the heart is divided into four chambers. Okay, so we can say that the right side of the heart, from which gets the blood, deoxygenated blood from all over the body, and from the vena cava, superior and inferior vena cava, the all the deoxygenated blood throughout the body comes into the right atrium of the heart and then goes to the right ventricle, and from right ventricle it goes to the lungs. It goes to the lungs where it gets oxygenated. Okay, and after the oxygenation, the entire uh, oxygenated blood comes to the left auricle, and from left auricle it goes to the left ventricle, and from left ventricle it circulates throughout the body by the systemic circuit. Okay, so we talked about the blood pressure. Okay, so the normal blood pressure, which is 120 on 80 mm Hg. Okay, okay, yeah, so we studied about so we know this is the normal blood pressure. So this blood pressure, the blood flows in our body with this pressure, this much pressure, only when it exits from the left ventricle and spreads throughout the body. Okay, so when the blood, so from the left auricle, the blood goes to the this left ventricle, and that time it undergoes diastole when the blood is entering. Okay, and then it undergoes systole. Okay, and when it undergoes systole, then with that what happens, the pressure gets generated. Okay, when via the systolic movement of the left ventricle, the pressure gets generated, and when the pressure reaches minimum 80, and then this semilunar walls gets open. Okay, so here we have, okay, so this is the diagram we studied in the last session, that it shows the ventricular uh, systole and the pressure, blood pressure in the left ventricle. Okay, so usually the blood travels throughout the body with a very low blood pressure. Okay, which is like 3 to 4 mmHg, when it is entering from the atrium, left atrium to left ventricle. Okay, then the blood pressure is very less, 3 to 4 mmHg. Okay, and then when the ventricle systole takes place, or when it initiates, so from here it initiates, and when the systole of left ventricle is happening, then the blood pressure is increasing. Okay, so blood pressure reaches, when it reaches 80 mmHg, that times the semilunar valve. Semilunar valve is the valve which is present in the left ventricle, which pumps the blood from left ventricle to the entire body. Okay, from the pulmonary artery. Okay, so at 80 mmHg, the semilunar walls open. Okay, and then again it goes up to 120 mmHg, and then it goes down to 100, and that time the semilunar walls again closes. Okay, so if your blood is receiving a certain amount of blood, not all of the blood gets removed or pumped in a one go. Okay, only 70 ml of blood um, in per beat removes from the left ventricle, and still some 25 ml or 20 ml bloods remain inside the left ventricle. Okay, so that much amount of the blood that is being pumped from the left ventricle is called the stroke volume. Okay, so and entire in the 70 ml, then we have 72 beats per minute. So entire minute, in a minute, the left ventricle pumps into approx 5 liter of the blood. Okay, so that is how the blood pumping takes place, and that is how what is we call our cardiac output. Okay, so the blood pressure, the semilunar walls open, and then again it closes, and again the pressure goes down, and then again in another ventricle systole the pressure again rises. So that is how the blood pressure changes in the left ventricle. Okay, so so before going to before we talk more further about the pressure, let's talk about the control of heart rhythmicity, the sympathetic and parasympathetic sympathetic nerves. Okay, so in the last class we have mentioned, and today also we have mentioned, that the heart generates the action potential by itself. Okay, and the region where the action potential is generated, that is the SA node. Okay, sinoatrial node, which is present in the right atrium. Okay, so there the action potential is being generated. Okay, and then from SA node it goes to AB node, and then it reaches throughout the ventricle via the bundle of His and Purkinje fibers. Okay, so how these points gets excited. Okay, so the signal these points are getting, they are getting from the brain. Okay, so from brain also, the medulla oblongata part of the brain, where the signal or the sense or the motor neuron gives the signal to the SA node. Okay, so in this medulla oblongata, you have a center of the nucleus. Okay, the entire uh, nerves that are coming out, that we call it as nucleus. Okay, this portion where the all the nerves are coming out. Okay, and that is present in the dorsal side, and it is a motor kind of nerve. Its name is vagus nerve. Okay, so it is dorsal motor nucleus of vagus. Okay, so this is very important. It is a dorsal motor nucleus of vagus, from where the origin of the vagus nerve is happening. Okay, so then the vagus nerve is originated. Okay, and then this vagus nerve again, um, so this is one nerve which uh, forms the ganglionic part here, and then again another uh, fiber again proceeds from that ganglion, which uh, reach localizes the signal to the SA node and AV. Okay, so before the ganglion, the type of nerve that is we are having, that we call them as a preganglionic fiber, preganglionic, and after the ganglion, the nerve we call it as postganglionic fiber. Okay, which is referred here. Okay, and also the signal is divided into two parts. We have a parasympathetic signal, and also we have a sympathetic signal. Okay, so parasympathetic signal is the one which is generated by the vagus nerve. Okay, which is generated at here at the medulla oblongata part, which is dorsal motor nucleus of the vagus. Okay, so here you can see from the brain, this vagal nerve, which is a parasympathetic system. Okay, it is bringing the signal from the vagus nerve, and before the ganglion, this is referred as preganglionic fiber, and then after the ganglion, the nerve which is carrying the signal, which is referred as postganglionic fiber. Okay, other than, and this is called as parasympathetic system. Okay, from the brain, from the spinal, from the brain, the signal is carries in the form of parasympathetic nervous system, and then the sympathetic signal, which is raised by medulla oblongata, but is not going directly to the heart, but it is being carried away by the spinal network, which is here, which is present at the thoracic region, which is T1, T2, T3, and T4, these regions. Okay, and then the signal is going and it is giving the signal to the almost the entire heart. Okay, and there also we have the ganglionic signal, and that also we have a preganglionic fiber, which is carrying the signal from the thoracic fiber to the ganglion, and from ganglion to the blood vessels and the heart. Then again it is carried by the postganglionic fiber. Okay, so here vagus or vagus nerve, which is part of the parasympathetic nervous system, which is carrying the signal to SA node and AV node here and here. However, the other spinal cord, the T1, T2, T3, T4, they carries the signal to the entire heart other than the SA node and AV node. Okay, and yeah, so okay, so as I have said that the signal arises from the brain and it goes to the heart, the SA node. Okay, so okay, so there also we have a certain type of receptors which is taking these signals. Okay, so the signal is also being or reaching to the brain or reaching to the heart in form of a chemical which is norepinephrine. Okay, and this norepinephrine is having a receptor here on the heart muscle, which is particularly the SA node, and this receptor is beta 1 adrenergic receptor. Okay, and this receptor is coupled with a protein which is called a G protein. Okay, so norepinephrine, which is nothing but a 7 transmembrane helical structure transmembrane protein, okay, which binds to the signal, which is nothing but the norepinephrine, and then it undergoes a conformational change, and which is coupled with a G protein. So norepinephrine initiates a cyclic AMP secondary messenger system, which activates protein kinase that phosphorylates. Okay, so the G protein activates the adenylate cyclase, which converts ATP to cyclic AMP, and this cyclic AMP has several functions, which activate several kinases, which phosphorylate several downstream proteins that helps in entry of the calcium that causes the depolarization of the cardiac muscle. Okay, and with the depolarization, the cardiac contractile force increases and the blood velocity also increases. Okay, so okay, so now we are connecting it with the type of receptor. Let us read. So the signal to the heart is coming into two series of neurons. Okay, one I can say, so if it from the brain, one neuron which is making a ganglion, then another neuron carrying the signal from ganglion to the heart. Okay, so it is two series of this particular neurons. So we have this preganglionic fiber, and after the ganglion we have the postganglionic fibers. Okay, and in this pre and post ganglionic fiber, the type of receptor, so the first neuron is referred as cholinergic uh, uh, receptors, and the second one is acetylcholine receptor. Okay, and at the heart, the SA node, the type of receptor that is present is a receptor which is nothing but, okay, it is a muscarinic receptor which is present in the heart. So all these receptor, I would say, they bind with the chemical which is acetylcholine only, but the type of receptor they are, that is being differentiated based on their binding to a certain kind of molecule which are present in the nature. So as I have mentioned previously, it is there are type of nicotinic acetylcholine receptor. So they are referred as nicotinic because they can bind to the nicotine. However, similarly, these receptors, even though they are also acetylcholine receptor, but they can bind to the a particular compound which is muscarine that is present in the nature, and due to that the name nomenclature has been done as muscarinic receptor. So in the heart we have this muscarinic receptor. So these receptor, uh, so the terminals are connected by the polypeptide chains that traverses the plane of the membrane 7 times, and the acetylcholine approaches to the receptor from the extracellular fluid and binds to the site of the receptor protein. G proteins interact with the cytoplasmic region of the receptor, and this M2 muscarinic receptor activation is coupled with the opening of potassium channels, causing potassium to flow outside of the cell, producing hyperpolarization. Okay, so um, okay. Yeah, so the potassium concentration is more inside and less outside. Okay, and so as due to this binding, the potassium channel opens and it removes the potassium from inside to outside. The more negative charge is being generated. So that is referred as hyperpolarization. Okay, more negative charges spread through the heart. So then the heart becomes more refractory to contraction and the rate slows, because for the contraction to happen, you need the depolarization of the membrane to take place. But due to the binding of the acetylcholine to this kind of muscarinic receptor, the potassium ion goes out, that causes the hyperpolarization, and which ultimately causes the heart rate to become slow. So these M2 muscarinic receptor cause action in the heart in the following manner. So in the SA node decreases the heart rate, AV node decreases the conductivity, and atrium also decrease in the contractility. Okay, so this is the graph which is showing the membrane potential. This is the control where you can see the heart uh, beads in form of membrane potential, and after the acetylcholine binding you can see, so in this particular time period in the control sample you can see 3 peaks are being generated. However, after acetylcholine binding you can see only 2 peaks are being generated. Okay, so the effect of acetylcholine on SA nodal cells is acetylcholine hyperpolarizes the nodal cell, thus it takes a longer time for the pacemaker potential to reach threshold. Okay, and the rate of rise of the pacemaker potential is also slowed. Consequently, the interval between the action potential is increased. And okay, so one more thing here. So as I have talked about this particular G protein coupled receptor, which is caused by the parasympathetic nervous system, which is generated at the vagus node and causing the release of the norepinephrine, which binds at the adrenergic receptor. Okay, which increase the contractile force. Okay, so there, um, so these beta 1 adrenergic receptor, they are being given a beta blocker. Okay, so beta blockers are beta adrenergic blocking agents, which is beta antagonist, and are a class of drug that are particularly used for the management of cardiac arrhythmiasis. Okay, protecting the heart from the second heart attack and the hypertension. So beta blockers cause your heart to beat more slowly and with less force. So if your blood pressure is being rising, okay, if you are facing a high blood pressure, then these beta blockers is used to treat that condition, which ultimately cause the heartbeat to become slow, okay, and with less force, which lowers the blood pressure. And these beta blockers also help open up veins and arteries to improve blood flow. And beta blockers block the action of the epinephrine and norepinephrine sympathetic nervous system. Beta blockers interfere with the binding of the receptor of epinephrine and other stress hormone and weaken the effects of stress hormone. So this is so electrocardiogram. We have this particular. So this is an ECG. You get such type of graph in the ECG, which is measuring the membrane potential in millivolts in the time manner, the real time manner. So this is what, this is nothing but it is showing the rhythmic contraction of the atria and ventricle with time in terms of voltage. So a normal ECG is look something like this. So what peak represents what is being shown here. So as mentioned already, the SA node will generate the impulse. Okay, so there the atrial excitation begins. Okay, or the, I can say, um so when the SA node gets excited, it ultimately allows more entry of the calcium. Okay, more entry of the calcium, and then calcium binds to troponin, which enhances the actin-myosin interaction, which overall causes the muscle contraction. Okay, and entry of the calcium ion causes the repolarization. So normal resting potential is in a negative. Okay. So our resting potential is usually in negative, and when the calcium ion enters, and it causes a resting potential to change from negative to positive. Okay, and that is referred as repolarization of the muscle. So when the repolarization happens, this particular peak shows the initiation of the arterial contraction here at the SA node, and then when it reaches to the AV node, then it causes a proper wave here, which is referred as P wave. Okay, and then again, so the impulse delayed at the AV node, which is referred with the P node, which is denoted at it with the P wave, and then the impulse passes to the heart apex, ventricular excitation begins. Okay, so the uh, intercept between two ventricles, there the excitation begins, and that is being uh, denoted with this decrease in the voltage. And then you have this peak, which is referred as QRS. QRS. So it is a QRS complex, and here you can see that the entire pulse has been spread throughout the heart, which is referred or denoted as QRS complex, and that shows the ventricular repolarization. And then after that, this is the peak, which is the T wave, which denotes the ventricular uh, uh, repolarization. Okay, so it starts with positive charge, then again the QRS also shows the ventricular depolarization. Then also we have this T wave, which shows the ventricular repolarization, again going back to the normal state, and then again the new rhythm of the start from the SA node excitation. Okay, so this is the electrocardiogram. So this is a normal electrocardiogram, okay, which you can see the PQRS, P wave, QRS complex, and T wave at a certain rhythm. Okay, by seeing a certain electrocardiogram, you can depict the condition or the rhythm of a person's heart. Okay, so this one compared to this one, if you'll see this, compared to this electrocardiogram, if you see this, you can see that there is a gap coming between each rhythm. Okay, between each rhythm there is a huge gap, okay, which is referred as bradycardia. So bradycardia is nothing but the slow heart rate, which is common in athlete. Vagal stimulus causes bradycardia. Okay, and then you have tachycardia. So this is sinus tachycardia. So tachycardia means faster heart rate, which causes increased body temperature, sympathetic stimulation, or toxic condition of the heart. So these two So here certain time intervals are being shown. Okay, the P wave should last for these many times, and the QRS complex should last for 0.08 second, and then the QT interval should be 0.35 second. So that is a rhythm with the time is fixed. So if your ECG is not following that particular time and those peaks are not falling in that time limit, that means there is abnormality in the heart. You have tachycardia, which is a faster heart rate, okay, or you can have a bradycardia, which is again showing a slower heart rate, okay, and this is a normal ECG where you can compare. And here again you are seeing uh, one rhythm, one beat, then again second beat, then again the arterial contraction supposed to start, but here it did not start, which shows that there is an SA block. The SA node is fails to fire, okay, and that's why there is a gap. Then again after some time it starts. So this entire thing shows SA block, which causes delays in the rhythm of the heartbeat. Okay, then okay. So this one is also, so this is a blood vessels dual intervention. Before that, talk about the uh, arteries and veins. So it is a blood artery and it is a blood vein. Okay. So artery carries the oxygenated blood, and the vein carries the deoxygenated blood. Okay. And they are made up of the smooth muscles. They are made up of three layers. Both are made up of three layers. So if you'll see the diameter or the thickness, okay. So the void between the okay, so the void between or the space between the artery is less, and the walls of these arteries are thick. So these are the smooth muscles, thick smooth muscles which is present inside the artery. However, in case of veins, these are the smooth muscles are thin. Okay, so it has three layers. Okay, first layer is tunica externa, then tunica media, which is made of the thick smooth muscle, and then you have tunica interna, which is filled with the endothelial cells. Okay, which releases certain kind of chemicals, for example, nitric oxide, which causes the vasoconstriction or vasodilation. Okay, that is a neurotransmitter. Okay, on the other hand, the veins are having a very thin, smooth muscles. Okay, so here the walls of the arteries are thick because they have to carry the blood with a lot of pressure. Okay, so before also when we have talked about the blood pressure throughout the body from heart to the lungs and then back to the body, back to the heart, and then again goes to the body in the entire circulation. So we have seen that the heart pumps the blood. Okay, okay, so here the ventricle pumps the blood to the these particular aorta with a very high pressure. Okay, it pumps with very high pressure. Okay, and then the pressure is dropped when pressure is dropped when it reaches or divided into the capillaries. Okay. So due to that, because they have to deal with a very high pressure, the wall of the arteries are very thick. They are made of very thick muscles. However, to carry the entire body's blood to the heart for the oxygenation, or like to the heart and then again to the lung for the oxygenation, the entire body blood needs to be moving through the veins. And to carry this more volume of the veins with where the pressure is not too much, the wall of the veins is very thick. So, and also the space is more so that it can carry more amount of blood. And also the arteries are having no walls in between them. However, the veins are supposed to have the walls in between them for so veins can be closed also, but the arteries should not be closed. Okay. So arteries doesn't have the requirement of being closed because the blood needs to flow throughout the body with the desired pressure. However, the veins, they are supposed to carry the blood from the body to the heart. So for example, time, time. Okay. So the veins are supposed to have walls. For example, the veins also carry the blood from your lower body to your heart. Okay. So against the gravity, it has to travel. Okay. So in that case, if the blood goes up, then it needs the wall need to be closed so that the blood which has moved up should not go back. Okay. So due to that, to maintain that, the veins also carry the walls. Okay, they can be closed and open. Okay? And before that, uh, this one also we'll read. So this is the heart, and this is the entire venous system which is carrying the deoxygenated blood, and the arterial system which is carrying the oxygenated blood. Okay, so the entire body, all the organs are connected with the capillaries, small capillaries which is carrying the blood. Okay, or doing the exchange of the oxygen and CO2 from the organs to the blood. Okay, and from the capillaries they form again form these vessels. Okay, which is which ultimately fuses with the venous system. Okay, and here it goes to the heart, and with the help of lungs it again oxygenated and comes back. So the again this aorta or the arteries which is conducting the blood again gets divided into the finer structure, which ultimately forms the capillaries. Okay, so before forming the capillary we have the pre-capillary sphincter. Okay, so these sphincters determine whether the blood has to go to the um, organ or not. Okay, or when desired, then only the sphincter will open and provides the blood to that particular organ. Okay, and here only the exchange, the capillary exchange, which is showing the exchange of the gases from uh, between cells and the blood, and then again it carries the blood to the venous system. Okay, and here only in the capillaries exchange we have the lymphatic capillaries. Okay, so the when the blood reaches to the plasma sorry when the blood reaches to the capillaries, what happens, the plasma of the blood is oozes out. Okay, so the plasma oozes out, and then it goes to the lymphatic capillaries. Okay, so lymphatic capillaries carries the devoid of RBCs, and it carries some WBCs and other portion of the plasma that excluding the large proteins, for example albumin, those cannot be passed from the capillaries to the this particular lymphatic systems. One second, please. Hmm. Okay, sorry. Okay, so we were discussing about the lymphatic capillaries. So the blood which reaches to the organ and in the capillaries, it oozes out some plasma. Okay, so in the form of plasma, it oozes out the ions and the other small proteins. Okay, and that is present in the lymphatic system. So lymphatic system is devoid of the devoid of the RBC. That is why it is also called as white. That is why it is also called as white blood. Okay, and yeah, and it is called as white blood. Okay, so these lymphatic system only, it ultimately mixes with the venous system. However, whatever fluid which is being gathered in the lymphatic capillaries, that is the one which forms the extracellular fluid of our cells. Okay, so our cells are surrounded by the extracellular fluid, and we discussed about it in the previous class also, that inside the cell there are certain chemicals or ions, and outside the cell, which is the extracellular fluid, also have certain ions or proteins present. So altogether, this extra lymphatic capillary, it forms the extracellular fluid. Okay, so this is the image which is showing the overall blood pressure throughout the body. So when the blood is so from entire body parts of the blood, it goes with 4 mmHg pressure, and it reaches to the right heart, and from the right heart it goes with a slow pressure only, which is 15 mmHg, and then it goes to the lungs, and then from the lungs it gets oxygenated. It again is with a 5 mmHg pressure, and the left heart it reaches, and there the pressure increases when the left ventricle undergoes systole. Okay, so there, the average pressure is 104 mmHg, and with that pressure, it distributes the blood to the entire body. So that is how. Okay. So this is the same. This is the graph which is showing the blood pressure in the different nerves or the veins or the arteries which is carrying the blood throughout our body. So here that 120 and 80 blood pressure that is arising from the left ventricle to the aorta. So here the blood is going with that 120 and 80 lower and higher blood pressure, aorta to large arteries, small arteries and arterioles, and then the pressure drops. Okay, and then when it reaches to the capillaries, again the pressure is around 8 mmHg, and then again it is some 3-4 mmHg, and then again in the pulmonary arteries the blood pressure is 25 and 8 mmHg. Then again it goes down, and again the entire uh, uh, system will again continue. Okay. So, so this is how the hemodynamics, the entire blood dynamics is taking place. If we'll see the question and answer from the uh, this particular part. So here it shows that label the following in the correct order. Okay. So this is the image of the entire uh, blood flow from heart, between the body, from heart to the lungs, and then the body back to the body. Okay. So the first one here, or let's say, so this is the one, third one. What is the third one? It is the arterial system or the venous system, which is carrying the deoxygenated blood.
Venous system.
Sorry,
Venous system.
it is a venous system. And the fourth one,
Arterial system.
yeah, it is arterial system. So venous system, this is arterial system. And then the second one here,
Lymphatic capillary.
yeah, this is the lymphatic capillary, where the plasma exchange between the blood capillaries and the lymphatic capillaries take place. Okay, which causes our, or which constitutes our the extracellular fluid. And the first one is the arteriovenous anastomosis. Okay, so that will be the correct one. And this is the wall. So this is the wall of our arteries. Okay, so here we have, as I have mentioned, we have three layering. So the second one is the outer layering, which is our tunica externa. Okay. The third one here is the endothelial cells. Okay. Third one, which is endothelial cells. Okay. Second one is the tunica adventitia, which is tunica externa. Fourth one is the smooth muscle, thick smooth muscles. Okay, which is nothing but the tunica media. And the first one is the tunica intima. Okay. This is the artery structure. Okay. So if we see these ones, this is the blood pressure in different capillaries or different blood vessels. Okay. So the second labeling where it shows the highest blood pressure. Okay. Which is what? So it can.
Skin.
Sorry.
Skin.
No. It is from the left ventricle when the blood pumps. It pumps to the aorta. Okay. So the second one is the aorta. Okay. Second one is the aorta, and then it goes to larger arteries, smaller arteries, and third ones are the arterioles. Okay, third one, and then we have the fourth one. Okay, where the pressure is, yeah, there the capillaries. Then from the capillaries again, the gas exchange takes place, and then again it goes to the venous system. So it starts with the venules, small veins, large veins, vena cava, and then from vena cava when it reaches to the pulmonary arteries. Okay, so there the pressure is found 25 mmHg to 8 mmHg. This is the pulmonary artery. Okay, so I think this one is the correct option. Okay. So I forgot to discuss one thing. Okay, so this slide I wanted to discuss. So here you can see these two images. Okay, so here you consider this one as a blood vessel supplying to the blood to the internal organ, which is nothing but the smooth muscle of the alimentary canal. Okay, so smooth muscle of the alimentary canal. So the blood, this particular blood capillary or blood vessel is carrying the blood to this particular uh, smooth muscle. Okay, which is part of the alimentary canal. Okay, so in the first image and from the in the both image, what difference can you observe? Can you tell me? So in the first image, you can see the lumen size is smaller, right? And in the second image, you can see the lumen diameter is, yeah, the lumen size is very large. So the difference between the upper image and lower image, if you will observe, you can observe these signal, right? So signal frequency is differing. So here the signal is uh, having more frequency, and in the lower image the signal is having higher sorry, here the signal is in more frequency, here the signal is lower frequency. Okay, here the frequency is more. Okay, when, so this says that when the alimentary canal, the blood vessel which is going to the alimentary canal, it is having more signals, which is getting more stimulus, it is its lumen size is getting smaller. Okay, it is undergoing constriction, and due to constriction, the blood volume or the blood which is going to that alimentary canal or that portion of the body, it becomes low. The blood supply becomes very less. Okay, because of the vasoconstriction. Okay, however, in the other case, the vasodilation occurs. Okay, so when there is less signal going to the alimentary canal, the blood vessel carrying to the alimentary canal, the dilation of the blood vessel happens, which supplies more blood to the alimentary canal. Okay, so there are two things happening. So when in an emergency, okay, in emergency condition, our sympathetic nervous system gets activated. Okay, so when the sympathetic nervous system gets activated, we saw which hormone is being released at the ganglionic level, norepinephrine, right? Norepinephrine. Also, along with that, we are also getting signal which of and the hormones which are being released are the adrenaline and noradrenaline. Okay, so all together the high intensity of the signal is being approached. Okay, so those signal when reaching to the smooth muscle of the alimentary canal, they are causing the vasoconstriction, which is lowering the blood supply to the alimentary canal. Because during an emergency, suppose you will having a flight condition where you have to run, your alimentary canal doesn't require that much amount of the blood. Your blood supply should goes to the your skeletal muscle, your brain, your respiratory system, because those body organs needs to function more efficiently in those frightening conditions. So that is why the signal is amplified, and those receptors that are present there that are causing the vasoconstriction, because there the blood supply is not needed. However, in the normal condition or in some other condition when the signal intensity is less, the blood supply is more to the alimentary canal because that is what is needed. Okay, during the similar condition what also happens, so the receptor, the type of receptor that are present in the respiratory system, okay, or in the skeletal muscle, the same hormones when act on those particular hormones, or when those particular receptors, okay. So this is alpha 1 adrenergic receptor which is present here in the alimentary canal smooth muscle. So which upon action of these hormones, epinephrine, sorry, adrenaline, noradrenaline, norepinephrine, it causes vasoconstriction. However, the same hormones which acts on the skeletal muscle, okay, where the receptor is different, so there they causes the vasodilation. Okay, because there in the skeletal muscle the blood supply is needed more, so there they causes the vasodilation, which carries more blood to that particular organ and increases the uses of that organ more in the frightening condition or in the emergency condition. So our sympathetic nervous system helps us in such a way, and it communicates with the different receptor in such a way that when the but one particular organ is needed, the blood supply towards that organ is enhanced, and the other organs which is not needed, your suppose your alimentary canal is not requiring the blood supply at that time, so there it causes the vasoconstrictions to lower the blood levels to that organ. Okay, I hope it is clear. If you have any doubt, you can ask me. If it's clear, there are no doubts, I'll conclude today's session. Maybe we can discuss in the next session if we do not have any doubt today. Okay, okay. Thank you. I'll conclude today's session. Thank you.